Us, they should be able to hear us. Cool. Yeah, sure. All right. Welcome, everyone. It's nice to have you here. Welcome to the 38th Annual Graduate Research Symposium. My name's Abby Repp. I'm a second-year PhD student here at Rubenstein. We're really excited to have 8 really great graduate student presenters today. Just a few little things. So we're going to be doing this hybrid. So we have, for people on Teams, you're currently in session 2, and everyone here, you're obviously in 103, but we also have a session going on in 311. So just a reminder that you are allowed, if you're interested in a presentation happening upstairs, you are allowed to switch sessions, but make sure you leave after the current speaker is finished. Also, speakers, you'll be getting a 5-minute, 3-minute, 1-minute warning to keep things on track. But other than that, we will be starting right at 5, so we make sure things are on track. So I guess without further ado, we can jump right into it. So our first presenter is going to be Ismar Biberovic. So Ismar is originally from Bosnia and Herzegovina. He got his undergraduate degree from UVM and is now pursuing a master's degree with his advisor, Professor Minnie Morales. So together they're studying dissolved organic matter and paleolimnology. So to kind of introduce you a little more to our speakers, we're going to do like a really tiny icebreaker between presentations. So this year's was, if you could be a hybrid of any 2 animals, what would it be? Because it's an amazing question. So Ismar would be a hybrid of a swordfish because it's fast and easier to get places, and an Arctic tern because it travels long distances. So Ismar, you're welcome to take it away.
So the next slide is the start of the presentation.
Yeah, you should be able to just hit the arrows.
Nice. Right.
Yeah, or you can use this clicker if you want. You just hit that arrow.
Arrows are fine. There we go. All right. So my name is Ismar. I study dissolved organic matter here in Vermont inland lakes with my advisor, Professor Morales here. So yeah, without further ado, I'm just going to introduce you all to what dissolved organic matter is for those of you who've had limited exposure to it. So dissolved organic matter, or DOM, is this heterogeneous complex mixture of various molecules. These molecules can come from decaying organisms, they can come from living organisms, they can be excreted by organisms. It also has its origin in these chemically or biologically degraded biomolecules. So it's really just a mix of anything or everything in the landscape. We talk about it because it's one of the most important sources of biologically available carbon to inland waters, but it also has a significant amount of nutrients that it delivers to inland waters, right? So this DOM pool right here, and a significant part of DOM pool is this dissolved organic carbon, which is the reason why most people study dissolved organic matter. So this dissolved organic carbon is further divided into non-humic and humic substances. Non-humic substances are these biomolecules that are easier to biodegrade, that are not as complex in structure. So, lipids, carbohydrates, proteins, amino acids, and such. Whereas these humic substances are just more complex, higher molecular weight compounds that accumulate in the landscape over time. So just like anything else, us humans have managed to alter the way DOM is processed in the landscape by altering the watershed, right? So we managed to alter the DOM exposure to light, making it more labile by applying nutrients, disturbing soil. We managed to also alter the timing it takes for DOM to reach inland waters. So DOM is not only terrestrially sourced, there's also aquatic sources of DOM. It is then photochemically, chemically, and biologically transformed, which is when we sample it, right? And this study relied on optical measurements of dissolved organic matter, and there are 2 types of optically characterized dissolved organic matter. There are chromophores and fluorophores, which is kind of shown right here. So chromophores are this bigger pool of dissolved organic matter, and then fluorophores are within that chromorphic pool of dissolved organic matter. So chromophoric DOM is very good at absorbing UV light and therefore heat. it kind of dictates, with that, it dictates how biogeochemical cycles and energy flow in aquatic ecosystems. So as we can see here, for instance, this is Abbey Pond in Vermont, down by Middlebury. And this pond has high concentration of aromatic dissolved organic matter, which appears more brown in color than usual lakes, right? And we classified this chromophoric DOM by using absorbance spectroscopy. So pretty much we emit light into our sample and see how much of it is absorbed by the sample. Fluorescent DOM is a fraction of this chromophoric DOM, and this fraction actually emits light back once excited with short wavelengths, right? And so pretty much this fluorescent DOM is very good at telling us more about sources of DOM, its molecular weight, aromatic content, and its freshness, or how bioavailable it might be. So pretty much what we did in our study, we sampled a bunch of lakes in Vermont. We used a fluorometer with some coding to produce this excitation emission matrix, right? So based on this excitation emission matrix, people have researched what different indices might tell us more about specific characteristics of dissolved organic matter. So using some mathematical formulas, ratios and such, people have found that some DOM indices are very good about telling us different things. Those things we focused on in this study are the freshness index, which tells us more about the contribution of recently produced DOM or how fresh it is. The SUVA, or specific UV absorbance value, which tells us more about DOM aromaticity, and then the spectral slope, which is an indicator of DOM molecular weight. So it's inversely proportional to DOM molecular weight. When it comes to our study lakes, we studied 97 different lakes in 2018 and 2019. 33 of these lakes were sampled in both years. We had 207 total observations, 113 surface samples, and 94 hypoliminetic samples. So every lake we sampled, we determined how deep it was, right? So if it was shallower than 2 meters or about 6 feet, we would only take a surface sample. If it was deeper than that, and if the temperature was equal throughout the water column, we also took surface samples. But if the lake was deeper than 2 meters and stratified, meaning that the difference between the surface water and the bottommost water, which we call hypolimnion, was significant, we took both surface and deep samples. And we also sampled our lake across different trophic statuses or productivity gradients. These are some of my lake characteristics. Watershed area to lake area ratio just tells us how much land there is compared to the size of that body of water we're in question. Elevational gradients as well. And then we also had some lakes that were very deep, like Lake Magog and Willoughby, which are up there. For statistical analysis, we utilize regression tree and random forest model. So we use them because they are very robust to missing values, non-parametrical data, highly skewed data and such. And our data was all of that. So we altogether reproduced 9 regression trees for all samples combined, so combined surface and hypoliminetic samples, for surface samples only, and for hypoliminetic samples only. So this is an example of that regression tree. We tried predicting the freshness index, for instance, in this case at least, using these predictor variables here. So what this regression tree tells us then is that watershed area to lake area ratio is the best predictor for freshness index. If it's higher than 11% or equal to 11% of the watershed, total nitrogen is the next best predictor. Otherwise, total phosphorus is. So you can just kind of follow this. And here's what we found. We found that spectral slope is negatively correlated to watershed area to lake area ratio. We also found the same case for the freshness index. And then for the specific UV absorbance value, we found that it's positively correlated to wetlands. However, if all of these are statistically significant, but R-squared value is quite low for the SUVA index. So to orient you with this plot, on the left-hand side here, we have only deep samples, only surface samples, and both of these combined here. And on the x-axis, we have percent variability explained by the random forest model. So the more variability we explain with our model, the better the model is, right? So what we found was that the main driver for DOM characteristics in the hypolimnion or in deep parts of these lakes was total phosphorus across the board, across all 3 different indices. For all these samples combined, we found that watershed area to lake area ratio best predicts spectral slope and freshness index, and again, spectral slope just tells us more about the molecular weight of these compounds. We also had negative values, which really just tell us that a regression model might perform better in predicting these three indices here. So with our discussion and conclusions, we found that higher watershed area to lake area ratio is related to higher molecular weight and more degraded dissolved organic matter pool. We also found that hypolenitic DOM is best predicted by total phosphorus, which possibly might indicate something about internal DOM sources in these lakes. And dissolved organic matter was less fresh and more degraded with higher watershed to lake area ratio, which indicates that these watersheds will allow more time for dissolved organic matter to be processed. And then this research would not be possible without Professor Morales here, but also Professor Clay Williams, as well as Vermont Limnology Lab, Vermont DEC Water Management Division, and then also Rubenstein Graduate Student Pandemic Fund, which I was funded on while I was doing this research. And that's it.
Wonderful. A full 1 minute before his 10-minute mark. Nice. So we have plenty of time for questions. So if anyone has any questions, please feel free to ask them. And I'm sorry for the Teams people listening. I forgot to mention this before, but as is typical of Teams, we're having technical difficulties with the chat. So if you have any burning questions, please send them to Caitlin Drasher's email. So cdrasher, DRASHER @uvm.edu. So that's it.
Go ahead.
So thank you very much for this presentation.
My pleasure.
What is the takeaway message for a Vermont resident who knows nothing at all about the research you're doing? What would you tell Vermont residents is the important message that they should understand from this research?
So we were to answer your question, we were hoping to find some land use drivers that drive DOM characteristics in lakes. We were not able to. One thing we did find is that preserving wetlands does make DOM less available to in aquatic ecosystems. So preserving wetlands is not only good for water purification, but also for dictating how bioavailable DOM is. And that's about it. We were hoping to find agricultural drivers and such, but we didn't. Franni.
What was the most challenging part of the study for you?
It was, initially we had this large data set, and we realized that that some of our numbers weren't, that there was an issue with an instrument that we used to measure dissolved organic carbon, or that DEC used to measure dissolved organic carbon. So we had to find those specific observations and remove them from our study because because the SUVA index uses dissolved organic carbon is normalized by dissolved organic carbon. So we had to identify those rows and get rid of them, which was a little challenging, but we did it.
Great. Well, if there is no further questions,
right.
Thank you, Ismar. Okay, and our next speaker is going to be Franni Hoag. So Franni is a second semester master's student in the Natural Resources program with a concentration in environment, society, and public affairs. Franni enjoys spending time outdoors, and her favorite outdoor activities are skiing, hiking, and running. Her research is on the Facebook pages of natural resource-based places in the northern forest states. And everyone's favorite icebreaker, if Franni was a hybrid of two animals, she likes to think that she would be a dog with wings. So without further ado,
Or a dog.
So you can just hit the arrows or space, or you can use the clicker too.
Okay. Okay. Hi, everyone. My name's Franni, as she introduced me, and I work with Pat Stokowski, and my research is on the Facebook pages of the natural resource-based places in the northern forest states. And I'm doing a comparative study of Facebook pages from many, many different types of natural resource-based places. So I have two research questions as of right now. One is, how do written text and visual images presented on the Facebook pages of resource management agencies use imaginaries to express their values of place? So I'm looking at place meaning mostly. And how do social media followers' comments on the Facebook pages of natural resource-based agencies in a region perpetuate imaginaries? I'm going to get...
Franni, could you speak up a little bit? Sorry,
yes.
I can't hear you.
No, it's okay. I have a soft voice. I'm sorry. So, as I mentioned before, this is a comparative study of Facebook postings. I'll be looking at both state and federally managed resource-based places. I'm building on prior research of public communications between managers and visitors and prospective visitors. And I am contributing to a series of studies about resource managers' communications. So, as I've said a few times already, managers of natural resource-based places use Facebook to communicate with visitors and prospective visitors. If you were to Google Green Mountain State Forest or White Mountain State Forest, one of the first websites that would come up would be a Facebook page run by the managers of that site. And this Facebook page kind of acts as a home website for these natural resource-based places. It often has contact information, location, hours, numbers that you can call, and managers who post posts about events that happen at these places. And what I think is most interesting about these Facebook pages is that people like you and me or anyone else in the world can comment on these posts and interact with the managers. And that's kind of what I'm studying. So I mentioned before, I'm doing research based on a series of social media related papers. And one of the first of which was done by Camille Marcotte, who was a master's student here a few years ago. And her paper, which was published very recently, was called Place Meanings in National Parks, a Rhetorical Analysis of Social Media Text. And so she looked at 3 major national parks in the country, kind of all over the country. There was no real regional focus to the study. And she looked specifically at the text posts. So she didn't look at images or comments. And she did a rhetorical analysis looking at the figurative language of those posts. So I mentioned in my first couple of slides that I'm studying imaginaries. And if you're anything like me, when I first heard this term, I had no idea what it was. So imaginaries are broad cultural, social, and spatial claims that groups of people collectively believe or assume to be true. And they often normalize social practices. They're all around us, and they're sort of this really interesting, obscure topic. And I definitely struggle with explaining it to people, but I'm going to try my best. Imaginaries are sort of central to tourism. They help perpetuate and cultivate the potential that a destination has. And they are especially rampant in social media marketing. Imagine you're on your phone sitting at home scrolling through Instagram, and all of a sudden a Instagram account for a resort pops up, and you click on this Instagram ad, and you're seeing all of these photos of a beautiful resort with people on the beach interacting with locals, and you start to form ideas about a place. But since you've never been there, you don't know if any of these ideas are based in reality. And that's sort of where the idea of an imaginary comes in. So our methods section is what I'm going to talk about next. Our data is going to be collected over 1 full year. I have started to collect some data, but not very much. We will be taking 2-week samples from each season, at least potentially each season. We're kind of still just trying to decide whether or not we want to include winter, because lots of parks in the northern forest states aren't open in the winter, especially any parks that involve water as a resource. Obviously, people aren't taking their motorboats out when the lake's frozen over. As I've mentioned before, we're going to be collecting text, images, and comments from these places, and we'll be performing a discursive and semiotic analysis on our data. So our study sites, as I've mentioned, are in the northern forest states. So this includes Vermont, New Hampshire, Maine, and upstate New York. We will not be focusing on New York City or Long Island. And we'll be focusing on the Facebook pages of the National Park Service, the National Forest Service, and the U.S. Fish and Wildlife Service, and the state managed versions of these agencies as well. We're hoping to look at a national park in every single state. So in Vermont, that is Marsh Billings Rockefeller. In New Hampshire, that's St. Gaudens, which is managed by Marsh Billings Rockefeller. We have yet to decide on our New York park or our main park, but we're probably not going to use Acadia. Acadia is very different than some of the other parks and places in this region because it's so large and so many people visit it every single year. We didn't feel like that would be a fair comparison. And Camille in her paper already touched on Acadia. So we feel as though probably we should pick a different park in Maine. We'll be looking at 3 national forests total. So White Mountains, Green Mountains, and Finger Lakes. Green Mountains obviously in Vermont. White Mountains are Maine and New Hampshire, and Finger Lakes is upstate New York. And we'll also be studying at least 2 state managed versions of these resource places in every single state. So here is a small sample of our data taken from the White Mountain National Forest on June 10th, 2021. I'll read it aloud to you. So it says, keep your eyes sharp because you never know who may be hanging around. The North American Luna moth may look like a leaf from a distance, but up close, he's one of the largest moths in North America with a wingspan of 4.5 inches. Generally a nocturnal creature, you'll be very lucky to see one in the daytime. So this was a post posted in June. It was accompanied with a few other photos, but I decided not to include them just because it would have made my slide kind of cluttered. And so this is kind of an average Facebook post, but what I thought was especially interesting about it was the comments. So this post had about 40 comments on it, and I would say for the White Mountains, that specific Facebook page, about 10 comments are normal, but this post had about 40, and they all looked something like this, where people were just really, really excited to share their photos of Luna moths. And I thought this was kind of interesting, and it kind of, I didn't see any evidence of Imaginaries or anything like that, but at least it showed that there's a community of people who are really interested in these Facebook pages and that interact with them regularly. And something I didn't include in my slideshow that I've noticed in looking at our data is that a number of people show up time and time again in these posts. Like they'll comment on multiple posts, so you know that they're very engaged with the page, and I think that's really interesting. As I mentioned before, we'll be performing a discursive and semiotic analysis of text, images, and comments from Facebook, and we hope to identify imaginaries and then use those imaginaries to help explain place meaning. So a discursive analysis is going beyond the sentence and going beyond the face value of the conversation to understand a deeper meaning. And semiotic analysis is the study of signs and symbolism. And that is all I have. I'd like to thank Pat Sikowski, my advisor, because without her I would not be standing here, as well as my committee member, my only committee member as of right now, Walt Consul. It's also been very helpful. Thank you so much. Any questions?
What are imaginaries again? I'm sorry, I can't wrap my head around it.
Sure. So imaginaries are thoughts or ideas that are collectively assumed and believed. So if you're looking through your data, what I found to be helpful is looking for words like I believe, or even most obviously, I imagine. If you see the words I imagine, that's a pretty good indicator that there's something there. They're also usually involved with emotion to a certain extent. They're very difficult to wrap one's head around. Yeah.
Would a good synonym be like a preconceived notion?
I think it's definitely similar. I don't know if they're synonyms exactly, but similar vein for sure.
And Seth, going off that, what do you have any expectations of what imaginaries you find in, like, especially like I feel like there's a common thread between the parks that you've chosen.
Yeah.
I can't even guess what they would be.
Yeah, I have a few thoughts. I think because just this region of the country. People have like an idea of the Vermont wilderness or the New Hampshire wilderness or the Maine wilderness as being well traveled but kind of rugged. So I'm expecting to see a lot of language describing that. And also if we decide to look into historic sites, I think New England history, at least I have a lot of thoughts about that. I did grow up here, so maybe that's an unfair advantage, but I think that'll definitely show up. I'm not, it's very hard to explain, and I hope I at least did an okay job, but yeah, any other questions? Okay.
So we have a few minutes until our next scheduled presenter is scheduled to present. So Sarah's going to go at 5:30. So to give people time from the other session to come down and switch if they want to see Sarah's, or if you guys want to switch to go upstairs, you're welcome to do that. And that gives people in teams time to do that as well. As a reminder for people in teams, if you have any questions, you're still in session 2. If you have any questions, feel free to email Caitlin Drasher at cdrasher, DRASHER@uvm.edu. So we're going to take, we're surprisingly ahead of schedule, so we're going to take just about 5 minutes so that people can take their time getting around. So, all right, let it go for 5 minutes. Yeah.
Alright, good to know. Yeah, because I remember
Cool. Caitlin, is that the only one that's remote? How dare he move across the you might not be able. He actually won't be able to see us. It's the last time he can pull like half my time. Yeah, he said he was going to set a timer. Oh, really?
I wish it was like in that corner.
If I can move it, it's not gonna
I feel like I stick to my time. I've been like. I'm trying something I'm surprised about that. It feels weird. I'm like, are we like that? Yeah. When I first started, it was like, or like first like ran through it, I was at like 12 Yeah. So Jen helped me because of that, but he only told me to add things. So I was like, I'm not going to be a 12, but I was like, that's kind of helpful.
Oh my god. Well, he's a puppy too, right? We do. And there they go.
Well, okay, I don't like if I like pause and then like 10 seconds over, so it's not like if I like forget my thought, then I'll do Now, I know. I feel like my project has so much background, so like just giving that is really helpful.
So unmuted? Yeah, I think it is. Okay, yeah. Cool. Yep. Okay, wonderful. Okay. All right, it's 5:30. We can get started again with our next presenter, Sarah Raimondi. So Sarah is a second semester master's student studying national parks and social media. And in her free time, she enjoys being outside and coaching lacrosse. And if she could be any hybrid animal, she would be a lion panda. A lion because they share similar hairstyles, and a panda because they only eat meat when they have to. So Sarah, you're free to come up here.
Thank you so much. That's credit to my mom, by the way. I did not think of anything. All right, let's see if I can work the clicker.
There should be the arrow on the right.
Great. So my name is Sarah. Thank you so much for that introduction, Abby. So this is gonna be pretty similar to Franni's presentation. I'm working on a series of papers with Dr. Zakowski on social media and national parks. So my research aligns with Franni's, but I have a different research focus and question. So my research question is, how do different types of parks in the national park system use Facebook to present their parks to social media viewers, forge relationships with the public through Facebook social media, and express and foster emotion within these posts? So I'm looking at the emotional response and the communication that these pages represent. So I'm going to start off with a little bit of context. There we go. So social media, if you don't have it, you know what it is at this point. It's growing exponentially. Everyone knows what it is, they use it. It can reach millions of people in an instant. So that's a really new, relatively new channel of communication because you're reaching a lot of people in a very short amount of time. Instead of like hanging up a poster or like having to go to the park directly, you're in this channel that's reaching a lot of people on a very accessible platform. So it provides the opportunity as a communication tool for agencies and participants for that instant interaction. And then looking at Facebook, Facebook is currently the largest social media platform, 2.5 billion monthly users. Lots of ways you can interact. You can like, you can comment, there's ways you can repost, you can follow pages and interact through comments and have discussion that way. Also, something that's interesting is the Facebook algorithm. It's kind of something that you can't really understand because it changes constantly, like it's changing as I'm speaking right now, and it's adapting to what people are doing online. But it's important for this study for a bunch of reasons. One, it's a limitation because I can't control the algorithm and what it does to the data and how I see data versus how someone else sees data. And also, when the algorithm caters to when you like something, your algorithm is going to adjust to that and be like, oh, Sarah likes national parks. I'm going to put more national parks in her feed. So liking things and interacting with things promotes that back into your Facebook page, and you're most likely to see it if you are following those pages. And then we go to the national park system. So national park system has about 8 billion visitors annually. It's not just national parks. They have over 400 different units, which I think is something that I definitely learned from beginning this study, because I was like, the national parks, it's the big ones. It's Acadia, it's Zion, but they have all these units, which is what I'll be looking into. So like monuments and historic sites and memorials and seashores. So as you can see from this map, they're scattered all around the country I'll be looking at regional ones, aligning with Franni's research and that angle of it. And the National Park operates on a dual mission to protect and preserve significant resources and then provide for the enjoyment of the people. That second part is really important because that extends to social media. When they're posting on Facebook, that's also aligning with their mission. They're trying to provide enjoyment for the people, and that's where emotion comes into play and makes an important impact. So, Facebook and national parks. I took this screen grab a week ago from the National Park Service page. Right now, they're sitting at 1.5 million likes. That ties back into my talk about the algorithm. So those 1.5 million people have interacted with the National Park Service. Their feed is going to reflect that, and they're going to see this information. So not only does their main page, but as I said, those 400 different units, they all have sub pages. So each one of those sub pages are going to have their own likes, their own people interacting with them, different content, and all of that. It's important to notice that previous research has linked visitors likely to visit social media after visiting national areas. I think that's really important because it shows that there's a connection between the two and that they're not mutually exclusive and that people go to parks, but they also visit social media and vice versa exclusively and together. So I just want to talk about some current literature. This paper is not about national parks, but I think it's really important because it talks about Facebook as a tool for communication and a place where dialogue can happen. This paper pretty much concluded that social media has immense communication tools, but they're not currently being utilized by agencies. And there are all these untapped areas of social media that agencies currently aren't using and that they're not getting the full range of response that is possible. And that organizations should capitalize on the dialogue aspects, the comments, the interaction that things like Facebook provide. I also want to talk about this study because their methods section is really interesting. This is kind of what I want to go towards as their table 6 in organizing how I'm breaking down information and collecting information. I'll be looking at texts, image, and comments. So looking to categorize those by what they are and how frequent they are on those Facebook pages. Second piece of literature, you've all already heard about a little bit today. Franni touched on it. My advisor, Dr. Stokowski, and her former grad student, Camille, worked on this recent publication, and my work is a continuation of it. So they worked on 3 major national parks. As you can see, Rocky Mountain National Park, Great Smoky, and Acadia, looking at text and images and categorizing the rhetoric, so the pathos, ethos, and logos of it. What they found from this study was that agencies mostly produce emotion-laden posts, so that pathos. And you can see that in the table 3, that top row, the 49%, 30%, 55%, and found that social media can really shape public view of resource places. And so my work comes into the conclusion of that paper. We're discussing that there are other national park units, so not just the big national parks like the 3 in this paper, but smaller units like seashores and historic places. So that's where my research comes in and extends on the series of papers that are being looked into on this. So my proposed methods. I don't have my parks yet, but there'll be 12 regional NPS units. I'm collecting text, images, and comments. Similar to Franni, it'll be in 2-week increments. March, June, September, December are the proposed dates. Also similar to Franni, we're not sure about the winter yet, depending on opening and closings of parks, but this is just where my head's at right now with how data collection will happen. I will be doing text analysis, so looking at keywords. So the word, what emotion is like the word love going to give you versus the words like can't and don't. And then visual data analysis. So that's looking at content of a photo, color of a photo, subject of a photo, and the emotional response that comes with that. And then the big question, why is this important? Why should we care about this? So this is really important. I think touching back on that first paper I talked about, that it's able to inform large organizations on how to use social media and how to inform organizations on the power and the emotional response that social media can have. And agencies can learn how to effectively communicate and use those resources and use their social media to the highest of their capacity and capabilities, and can promote a positive emotional response back to that national park mission. That's what they're trying to do, connect people in a positive way to natural spaces, and you can do that while not actually being natural space by being through a computer screen, which is really interesting. And promote constructive conversation. Comments are really cool. It's direct and immediate, and you're interacting with not just the agency, but the people that are also commenting on it with you. And questions? Yes.
This might be a question for both you and Franni, but when you're doing this research on Facebook, do you have to make a separate account, maybe from if you have your own account, in order to look at the comments and see things that aren't, I don't know, biased by your own algorithms or things
Yeah,
like that?
so I actually haven't, that is a great question. I haven't started collecting data yet, but my advisor and I were talking about how just data collection will just be done by me. My advisor won't be collecting data, no one else will be collecting data, so that it's just my Facebook algorithm. Also, it's important to note that the Facebook algorithm is really important on your feeds, but if you go look at Acadia National Park and I go look at Acadia National Park, we're gonna see the same thing, unless your friend happened to comment on the post, then it'll be at the top. But other than that, if I'm just collecting all the data myself, it shouldn't be very biased. Yes.
Have you also looked at what it might like for international users? Because I know Facebook is very different in America than versus India, and how the algorithms work there, and it's very difficult to regulate certain facts.
Yeah, that's a really good question, and I'm going to be totally honest, I have not thought of that, and now I definitely will. So that was actually really constructive and helpful. Thank you very much. Ismar.
I'm very curious as to how you all analyze comments. Like, is there a way to export them and see how many times a word is mentioned, or like, do you just have to go through every single comment and count the word?
I'm still in the discovery phase of looking through literature and looking through different ways that we can go about that. So working on it.
All right.
When I know the answer, I'll get back to you.
What are some of the ways that you plan on looking into how emotion is conveyed via post?
Yeah, so like text or image or both. So in the text, I definitely like word heavy words that people are using. There's a paper that I've done a bunch of research on called #ILoveLondon. It's all about the power of the word love. It's like, that's a pretty clear emotion that you can have. There are like more complicated ones, obviously. Some words can have different meanings to different people. And then images that's a little tricky, looking at color. I think definitely composition, subject. Yeah, definitely something still exploring, but hopefully, yeah.
This is a busy question. But
Yeah, no worries.
I saw, I'm not sure if you mentioned this or not, but how did you select the specific spots that you're analyzing? Because I saw it was like the Eisenhower homestead. And
yeah, those screen grabs I meant to say were probably
okay.
misleading because I don't have my parks yet. Sorry, I meant to say that, but then in my brain, presenting. I don't have my parks yet. Those were just some examples of different sites and what their Facebook pages look like.
Got it. Okay, great.
And you said it's going to be regional? It'll be
regional, yeah, it'll be in New England.
Any other questions? All good? Well, thank you so much, Sarah.
Thank you all.
Perfect.
Okay. No email questions? Okay, good. I keep forgetting to check in on.
No, you're fine.
Okay. So our next speaker is Daphne Zencey. So Daphne is a native Vermonter. She grew up exploring the forests and lakes surrounding Calais, creating an unbridled passion for the outdoors. After a stint in the U.S. Coast Guard, she dove into academia, completing both a bachelor's, a BA and an MA in art history. Eventually, she became restless and realized her true passion lay more.
Person over here.
Oh no. Darn. Okay. She realized her true passion lay more heavily within the natural world, landing her here at Rubenstein. In her spare time, which
She put quotes around. In her spare time from school, she enjoys mountain biking, fly fishing, uphill skiing, yoga, cooking, and travel. So Daphne says that she is most certainly part red panda and part wild horse.
I didn't really explain it.
No explanation needed. Exactly. So
Bolt out of this.
Uphill skiing.
Uphill skiing. Yes.
Oh, nice.
It's fun and what's the phrase that we sufferfest. So you ski up the mountain and then you ski down it. Anyway, this is not my research topic, but I can talk about that probably for longer than I can talk about my research topic at the point of my research. So I am doing presenting place through stories, evaluating the websites of AFN entrepreneurs. And as I mentioned, this is I came up with, and Patricia Stakowski is my advisor, and it's been sort of a rocky path to get to this topic. And I think I finally landed on this maybe 3 weeks ago. So it's still sort of in its zygote stages. But that being said, it's very interesting and I have plenty to show you. So the purpose is basically to figure out how, similar to the Franni and Sarah actually, like how do websites convey a message to the broader public? So how do websites used by alternative food networks convey and enhance the meaning of people and place to this broad audience? Again, where millions of people can visit these websites on any given day, any given time. And in order to get started, I need to explain what AFNs are. So alternative food networks are commonly defined by attributes such as spatial proximity between farmers and consumers and fair trade. So in our Burlington community, greater Burlington community, we have places like the Burlington Farmers Markets, we have our local CSAs, dozens of those available. We have farm stands, and then actual agriculture like the Intervale and the small farms that are supported there. So all of those entities are considered alternative food networks because they are the alternative to, say, your Shaw's, your Hannafords, your Price Chopper. And speaking of Price Chopper, I don't know if you guys have been here for a while, but the Shelburne Road Price Chopper recently rebranded itself, and I think this is across the board, as Market 32 in an effort to sort of not dupe the customer, but to try to rebrand themselves as more of an aesthetically geared more towards the farm stands and farmers markets. And you go into that space now, and it definitely feels more welcoming, more engaging, and there's something in that as far as conveying place and space and how that relates to us as consumers of food. So I mentioned place. What contributes to place? Again, very succinct definition is basically the physical characteristics of a community, how it's designed, planned, developed, and then maintained, and then how that place then affects the quality of life in the people that both live in the place and then visit or plan to visit in the future. So again, it's sort of like Imaginaries, I think, I don't know. Yes, but it is this idea, well, place itself is very structured. So here we have Church Street, again, it is the Church Street Marketplace, the buildings, the church at the top, that conveys Church Street as a place. However, what this definition doesn't include is intangible qualities. So again, more sort of like the Imaginaries. So the definition of the physical attributes of quality of place doesn't account for these intangibles that really are part of the essence of a place. So what is the essence of a place? Things such as the vibrancy, authenticity, and distinctiveness that are experienced by both the users, so those who live here, and then people who come and visit, and then the users of that place also then contribute to the essence of the place. So us all combined as this community of Burlington help create the essence of place that is Burlington, Vermont. So my research question is going to focus on how do our local alternative food network entrepreneurs in the greater Burlington area convey this concept of place via their website presence. And as Sarah did, I just sort of screen grabbed 3 options. I will or will not use these, but again, Burlington Farmers Market, the middle one is Philo Ridge Farm, and the bottom one is Jasper Hill. And I'm sure we've all visited websites before and noticed.
You might have toward the computer.
Oh, up into the sky. All the websites have these about us or our stories. There's always something on the websites that is there to compel the visitor to like click and learn more about what this business is. But why? Like, what is it about the stories and the about us that is part of their marketing strategy, basically? So alternative food network entrepreneurs can utilize these areas as a way to convey the quality of place to a variety of audiences. So here I have the Pete's Greens. I'm sure we're all familiar with Pete's Greens, and his about us is pretty comprehensive. You're not supposed to be able to read that, don't worry. And again, just how these across the board, all these alternative food network entrepreneurs are using their websites as ways to create this concept of place. I particularly thought Jasper Hill Farm had a really interesting, so this is their landing page when you go to their website. I don't know if you can see this, it's a little pale, but right there in the middle it says a taste of place. And so they're specifically branding their cheese as being able to taste place through consuming their cheese, which is just like, I need to figure this out some more, but it's really cool. And then along with their, their about us, our story, I'm also going to focus on the imagery that tends to come with these websites as well. And again, a lot of the same images come up across the board, whether it's on websites, social media, but it's like the iconic rolling hills, red barns, cows in a field, sheep in a field. And again, I pulled these from a lot of the websites that I've been looking at. The Intervale there in the middle, Jasper Hill Farm, Philo Ridge. So Philo Ridge Farm is both a farm, an eatery, and a market. So that's their showroom there. So again, they have these on the landing pages as promoting this aesthetic of what place is along with the story. Again, you're not supposed to be able to read all this, but I just wanted to pull a section from Philo Ridge. Has like 3 tabs under their about us. There's about us, farm history, and then the agriculture that they do. But I thought this was particularly interesting just as far as you can see the depth of text that they're promoting in order to convey the sense of place. And at the very bottom, it ends by saying, we strive to produce and share nutrient-dense foods, provide an exciting learning environment for our visitors and staff, strengthen our vibrant local community, all while honoring the integrity of the land. Food brings people together. So again, I'm going to go into further depth in researching this, similar to what Sarah I think is going to do, like do some text analysis. Actually, that's my next slide. Methods. So I'm likely going to sample approximately 20 websites from the greater Burlington area. And then I'll study the content of these about us, our story sections for those key phrases, those keywords that tend to pop up again, that convey the sense of place, as well as imagery used, again, the aesthetic value of story and imagery. And from that, I plan on developing a discourse analysis about the ways that alternative food networks communicate with the public about their specific place and what they're bringing to the marketplace, basically. So why does this matter? Well, first of all, I just think it's really interesting, like how people consume food, where we get it from, our relation to food. But that's or just my own interest. But understanding how these entrepreneurs describe their story relative to their brand and the product will provide future scholars, future researchers, a place to launch from. But I think more importantly, it'll be really interesting for marketing purposes, especially like the brand of Vermont, although this is just the sample studies space, but just how they are branding themselves via their websites, via their text, and via their images. And just a happy little landing spot. Again, some of the words that I just found popping up a lot, nothing shocking here. Food, community, sustainability, place, mission, healthy. So these are the kinds of words that I will be looking out for across the board of all the websites. And that is it. Thank you. Any questions?
I have a silly question.
I love silly
The
questions.
last slide that you just did, what do they call those?
Word clouds.
Word clouds. Thank you.
Except I'm in my Vermont cloud. Oh, yeah. It's a great question. Anybody else? Steve?
Yeah, I was thinking about farms and their different approaches, whether it's a real urban approach, Burlington area, or a real rural approach. And are you going to look at a little bit of like their website and their marketing based on who they're trying to reach? If they're trying to pull a Burlington crowd, or if they're trying to pull a, like a tourist, tourist agro farming interest or something.
Yeah,
Stowe Farmers Market.
right.
Right.
Yeah, I think I need to definitely consider who the broader audience is. For example, Jasper Hill Farm is clearly marketing outside of the state. They're looking for the New York dollars, the California dollars to spend, and that's why they're branding as taste of place. Like, this is the most authentic experience you can have. Buy our cheese, spend the not thousands of dollars, but we're very lucky that they're in our backyard, basically. But yeah, I think that'd be really interesting to sort of try to figure out what the demographic is as far as who the website, because like the Intervale is probably geared more towards the local communities here, maybe reaching into like central Vermont, but for the most part, it's a localized movement. So that's a good point, definitely.
Sorry, I'm so unfamiliar with this field, but like, are you allowed to talk to the people who make up these like the people that chose the words?
So this was originally the study was going to be more geared towards personal interviews and actually talking and doing the data collection that way. Unfortunately, I think just time constraints and going through the human subjects. I personally would love to talk to all of them. So maybe after I do the study, I can do that without any conflict of interest. But for this study, I'm just studying the content that they choose to put up on the websites.
Because you have to go through IRB if you're going to talk to people.
Right.
Yeah.
Someone else had a question?
I did, and I could have asked any of the three prior presenters this as well, but you mentioned that this definition of place kind of excludes these like factors of vibrancy in the people. So when you're looking at these websites, I know you went into it a little bit, but especially when you're looking at something for example, vibrancy, what are you looking for?
I haven't figured that out yet, but I will. Yeah, I think that's going to be a tough one because it is sort of this concept of like imaginary. What is vibrancy? So yeah, I'll look into that. Okay.
How do you pick places that you want to study?
I'm going to do whatever Patricia tells me. She and I'm sure will discuss and figure out. I would love to do some broader range, not just like Burlington direct, but there has to be a cap at. And I want like a broad variety, obviously, as well. Not just farmers markets, not just cheese makers, like maybe some beer makers, wine makers. I get to study all the really cool, fun things that I love to consume.
Yeah. I was wondering, for vibrancy, do you think, is there a way to quantify colors used on pH?
There's, yeah, definitely, and that, I think the reason Dr. Sikowski and I came to also include the images is like my art history background. I definitely have that, like, that knowledge base and that interest in like how do images convey a meaning. So I think there's definitely something about like how many yellows are represented versus blues and dark colors. Yeah, I'll think about that definitely. Is that it? Okay, thank you. Thank you.
All right, and Daphne was at 10:00:00 on the timing, so you nailed it. Exactly. Okay, so our next presenter is going to be Steve Bartlett. So Steve is a master's candidate in natural resource and aquatic ecology in Watershed science program. He is in his second year and is in the middle of his thesis research in which he is studying management techniques for the invasive reed canary grass. I'm not going to try to pronounce the scientific name for that. And its effect on mortality of newly planted trees in riparian floodplains of the Lake Champlain Basin in Addison County, Vermont. And if Steve could be the hybrid of two animals, he would be a brook otter. So the combination of a brook trout and a river otter, so that he could live in the same rivers with other fellow brook trout and otters. So without further ado, Steve, take it away.
Make sure this works here. Yep. Okay. Yeah, so my research, the idea originated in as we're looking at ways to work on the water quality of our rivers, lakes, and in particular Lake Champlain. One of the questions was, what's a common practice out there that we can identify that's a nature-based solution to address that? And that's obvious is one of those is establishing riparian buffer zones. As we looked at those riparian buffer zones, the question came up of what is a problem within establishing those buffer zones. Why can't we just go out and establish those and create forests along rivers and help reduce the amount of phosphorus and nitrogen and pollutants and sediment and all the things that can get into our waterways? And what is the problem that's making that more challenging? And in talking with a lot of the practitioners and the people that are on the ground doing this work and planting the trees and doing the research and trying to figure out what these problems and issues are, one of them became known as an invasive species called reed canary grass that is very problematic in the establishment of newly planted riparian buffer zones. And in those riparian buffer zones, reed canary grass is an extremely quick growing grass that can reach up to 5 and 6 feet tall with a very dense root structure. And there's a number of ways that reed canary grass can inhibit the growth of tree stems, a myriad of ways, in fact. One is obviously outcompeting the newly planted trees for shade, soil, for nutrients, shade, and light. Light by being shaded because of the quick growth. The other is after it grows to be about 5 or 6 feet, in the fall, and when it dries out, it can create these big clumps that in the winter create ice and can break off stems. That's another way that it can cause mortality in newly planted stems. And the other is that some of those kind of teepees with all the grass matted up against the stems creates this really very nice hospitable home for things like moles and voles that like to eat all of the bark and girdle the stems, therefore really increasing the rate of mortality of the stems. So that is the problem. And the question is, how effective is tilling, mowing, and herbicide use at managing the growth of reed canary grass, and what is the resulting effect on mortality of newly planted stems? Currently, a common practice nationally, but also in the Lake Champlain Basin for controlling reed canary grass is the use of glyphosate, which is a Roundup type substance. There's there's been studies on different uses of glyphosate and then also different ways of tilling and mowing in various ways of trying to control reed canary grass. But currently one of the most effective is glyphosate, and that is either being applied in larger quantities for entire areas and being sprayed in the fall prior to planting in the spring. So it's the scorched earth mentality, creating a sterile seed bed for your planting in the spring, right? Another way that's common is the area gets mowed off, trees are planted, and then a conservation crew or a nonprofit or a profit company goes in and sprays glyphosate around the tree in about a 6-foot radius, and it allows the tree to grow up without the obstruction of the reed canary grass. Both of those are somewhat effective in the research, but also create that problem of we are using an herbicide in a floodplain area where we're trying to improve water quality, and even though the half-life of glyphosate is 45 days, it's assumed that it's not going to have a huge impact. It is still adding an herbicide to our environment, and one, lots of landowners and practitioners have no interest in herbicide application on their property, and two, it also, has effects for amphibians and animals and plants and the individuals and persons that are applying it, right? We all know that that is shown to be an actual carcinogen. So my study, we have 8 different sites across Addison County, Lake Champlain Basin, and those are all in wildlife management areas. They all have clay soils in floodplain areas. They needed to be somewhat accessible to a tractor so that we could mow and till. I'll talk a bit about that in a moment. And then also they had to be just above the floodplain enough so that in flood and scouring events with ice, that didn't shear off all of our stems and basically reduce all of our, increase our mortality to the point that we no longer have any stems to count. So those sites are Little Otter Creek, north and south, up in Ferrisburg, Otter Creek, Lower Otter Creek in Virgins, Dead Creek in Addison, Whitney Creek is down by the Champlain Bridge, and then Lemon Fair is in Cornwall, down by Middleburg. This is just a quick map of some of the soils, these being our sites. Most all of them, with the exception of one, fits right on the edge of a sandy loam and a clay soil. We also are doing soil samples to see if there's any relationship between what we find in the soil samples. Oops. Here's a quick picture of one of the sites. This was right after we mowed. So in this site, our control will be the common practice right now for reed canary grass, and that's the herbicide. So this will be herbicide, and then our treatment will be tilling and mowing. We'll talk about that in a second. Divided up each site into a control and a treatment with 9 subplots within each side. This will identify it more clearly. So here's our treatment. We mowed it, we herbicided it, we tilled it with a power tiller to 8 inches depth. Then we put herbicide down again, and then we tilled it. This was all in the fall of last year. The control was mowed, tilled, and then tilled again. And we had about a 10-day to 2-week period in between herbicide and till applications to allow the herbicide and the tilling to do its work. So within each of these subplots, we've got 10 native stems per subplot. The subplots are, let's see, the plots are 0.25 hectares each to make half a hectare in total, which basically gives us a planting density of 1,440 trees per acre, which is actually higher than what a lot of the normal practitioners are doing for a planting density. But we really needed the number of trees per subplot to have statistical significance is why we have so many trees per acre. I gotta keep going there. Whoops, I'm going the wrong way. Sorry. So here's a quick rundown of our sites. So 8 sites, 72 control, 72 treatment. We also estimated the percent of cover of reed canary grass with a meter by meter quadrant. Place that in every subplot and estimate 10%, 20%, 30% reed canary grass. We did that once a month for the month of June, July, August, and September. Okay. Oh, and then also we used a manual weed eater each month in the treatments to basically mimic mowing. And then we also mowed the control twice to mimic mowing as well, and some sort of input by the landowner as well, instead of just herbicide only. And that's an electric weed eater, which was pretty cool. I had like 6 batteries and a battery charger in my car, and it was pretty awesome. Special thanks to my planting crew and my advisor, Chris Stepanek, who planted a lot of trees with me. Thank you. This is a quick picture of in June, one of the trees that made it. Here's one that didn't. That's what it looks like right after we planted. You can see that's the herbicide, that's the non-herbicide. That's June. Herbicide, non-herbicide. July, you can see some other invasive species in the herbicide section coming in. August, you can see the trees grew a little. And then September, quite a bit of growth. The most interesting part of this in my last few seconds is in July, you can see that outside of this plot, the reed canary grass is 4.5, 5 feet tall already. So without anything, that's what we're looking at, and that's why we're doing this research. My partners are here. It was pollution prevention habitat grant from Lake Champlain Basin Program and the Watershed Forestry Partnership, UVM Extension, and Lake Champlain Sea Grant. Thank you. All right, questions? Yes.
I noticed that your half-meter hectare sections were they immediately adjacent to each other?
They were. They're immediately adjacent to each other, and we put a barrier in between to prevent any sort of leachate of the water and the, yeah.
What was that barrier made out of?
It's made of basically a weed material.
Okay.
Yeah, and so possibly some of that herbicide could have leached through a little bit, hopefully not that much, but also the weed barrier prevented reed canary grass from the rhizomes really moving was what we were really concerned with. Yes.
Have you analyzed any effects from the ability of the planted trees in the herbicide versus non-herbicide for the mowing area, like the success rate?
Yeah, so so I probably skipped over probably one of the most important parts of this is that we are counting live and dead trees for each one of those subplots. And so each month we went out and counted live, dead, took pictures of each subplot. I even took pictures of the dead trees. If we could identify the dead stems, I identified them as well. So I haven't done all the statistics on it to figure any of that out yet. We have a whole another summer of data collection where we'll do the same thing. And that summer is really critical in the growth of stems, is our second and third summer usually. But, you know, anecdotally, red maple and silver maple trees appeared to be dead after 2 months, but then got stem growth after the 3rd month in August. They started growing from the stem. And bur oak trees didn't make it through the really dry June. Not all of them, but if there was one species that struggled, it was bur oak, and I'm not sure why, other than it was super dry in June.
Any other questions? How about our team's chat? Anyone sending any emails?
Well, the chat's still broken, but I haven't gotten any emails. I keep checking my phone for emails.
Oh, wonderful. Well, thank you so much, Steven. Great. So we're right on time. So our next presenter is going to be Maggie Batton. So in addition to being a master's student at UVM, Maggie works for the United States Department of Agriculture. Her research is looking at improving oral rabies vaccination strategies in urban areas, and is a collaborative study with UVM, the USDA National Rabies Management Program, the National Wildlife Research Center, and Vermont Wildlife Services. And if Maggie could be a hybrid of any two animals, she would choose to be a cat and a bird, because she could be extremely nimble on land and also be able to fly anywhere she wants. So Maggie, take it away.
Okay, so which button do I put? The arrows here. That's actually a laser pointer, which I probably should have told you.
Oh,
But
okay. All right. So today I'm going to be talking about improving the success of rabies vaccination strategies in wildlife across the greater Burlington area. And this project, my advisor is James Murdoch. Okay, so you most definitely have heard of rabies before, but you might not know that it is a zoonotic disease, a fatal zoonotic disease, meaning that it can be transferred from species to species and to people. It's spread through the bite or scratch of an infected animal, and although it is completely preventable, if it's not treated before symptoms arise, then it is always fatal in both wildlife and people. So this is one of the world's most deadly diseases, making it a major global public health concern. So countries from all over the world have banded together to try and eliminate dog-mediated rabies virus by 2030. So the canine variant has been eliminated in North America, but we still have the highest diversity of rabies vector species globally. And although very few people die from rabies virus in the US, about 55,000 people per year are still exposed to this virus. And in the eastern United States, the raccoon variant is one of the most major sources of exposure. So the USDA National Rabies Management Program was created to manage rabies virus in the US, and specifically the raccoon variant in the eastern United States. So they have two main goals for this program, and the first is to stop the spread of rabies outside of areas that are enzootic to the disease, and then to vaccinate a sufficient percentage of the target populations, which is thought to be about 70%. So this is done through a program known as oral rabies vaccination, which involves distributing baits on a landscape that contain the liquid vaccine that are covered in an entractant. And the idea is that the animal will bite into the bait, exposing themselves to this vaccine. Ooh, this way, sitting here like this. Okay, so there are several different baiting strategies to get these baits out on the landscape, depending on the type of or the level of development in the area. So the most common method that's used in rural and forested areas is fixed wing aircraft. Helicopter baiting is used in more suburban areas, and hand baiting is the most common method that's used in urban environments. And then there's bait stations, which are less frequently used, but they are a method that's used in urban and suburban environments. So there are many considerations that need to be taken into account when planning for ORV. For one, bait density is a really critical factor because it needs to be able to reflect the species population density in the areas that we're targeting. But there are several factors that actually limit this ability to bait at this density in real time, such as increased amounts of roads or sidewalks. So the National Rabies Management Program created an algorithm to estimate how much of an area is deemed unbaitable. And this is referred to as the off-time calculator, which adjusts the target number of baits per cell based off of baitability. So basically, if there's increased off time, then there's going to be less baits in the landscape. So ORV has been shown to be really successful in these more rural settings, but the RVNA percentages are significantly lower in many urban areas. And there's a lot of questions about why this is. But baiting in these urban environments do come with many challenges. For one, there's an abundance of alternative food sources. There's a chance of non-target bait competitors, and there are some hand baiting limitations because this does occur mostly on roads and sidewalks. So we might not be accessing some of this suitable habitat. So there have been several studies that have been done trying to look into some of these potential factors, but there are still a lot of unknowns about this issue. So this is a map of the current operations in Vermont. The map on the left is, so anything that's shaded orange is where we conduct or where we distribute bait baits. And this is done mostly through aircraft. But in our more urbanized settings, we do use hand baiting. So on the right-hand side, this is a zoomed in view of the Burlington baiting. So anything that is a grid is where we distribute our hand baits. And then the orange shaded area around that is our newly implemented helicopter zone. So historically, hand baiting has been the only ground baiting method that's been used here. So in our rural settings, we've been seeing really a really high success of these programs with high RVNA conversion rates. But in our more urban areas like Burlington, specifically Burlington, these rates are significantly lower, which is shown on this graph. So we have tried refining the hand baiting strategies to get a more even bait distribution across the area, but it's still largely restricted to roads, and we haven't seen an increase as of yet. But Burlington is still a major study area for multiple rabies research studies that have occurred over the past couple years. For one, but yeah, despite these studies, there's still a lot of questions that are left unanswered about this area. So that's where my study comes in. I have two objectives to my study, and I'll walk you through each individually. So the goal of the first study is to improve the effectiveness of our current ORV baiting strategies in these urban areas by trying to understand what factors are driving this bait survival on the landscape. So I have 100 cameras total that I've been distributing across the Burlington area in our ORV zones. And so 80 cameras are within the highly developed and moderately developed areas. And then we have 20 cameras that are within the low development helicopter zone. So these cameras are going to be monitoring our on-rab baits that we distribute within Burlington, and I'm going to be mimicking our current hand baiting strategy to try and identify what is actually happening to these baits once they're on the landscape. So my specific objectives are to monitor the fate of on-rab vaccine baits in real time as we're distributing these baits in our operations. I'm also going to be summarizing the fates of baits from the data that I collect with my cameras, and then I'm going to be modeling the probability of bait uptake. So I did just conclude my first field season, so my cameras were out for a maximum of 4 weeks from the date that the bait was deployed. And I had weekly site checks where I recorded the status of the bait as one of 4 options, being it whether it was taken from the camera site, it could be partially chewed and but still viable, or it could be whole and present and whole, or it could be fully chewed and non-viable. So the data is going to be summarized by calculating the proportion of sites as bait loss, which I just showed you, as well as the cause of bait loss, the average persistence of baits, and my development categories. And then I'm going to be modeling the probability of bait uptake by a target species over time using a logistic regression generalized linear mixed effects approach to evaluate how different site level and landscape level factors influence this probability. Okay, so I'll move on to objective 2, and the goal of this objective is to assess the effectiveness of adding our bait stations to hand-baited areas at improving the seroprevalence in these urban environments. Specifically, I want to quantify the effect of adding bait stations to hand-baited areas by comparing results with our hand-baited only areas. So I have 2 treatments in my study, the first being bait stations added to hand-baited cells where off-time is greater than 50%. The second being bait stations added to hand-baited cells where off-time is between 30 and 50%. And then I have a control group, which is my hand-baited cells only. So I have a total of 16 cells, and 2 bait stations are added to each of these cells to increase the off-time, to increase the bait density to absolute density, and to try to give these animals another chance to encounter this bait. So all of my vaccines have unique molecular biomarkers that are used to identify where the animals ingested the bait, and this is found in the blood serum of these animals. So all of my hand baiting baits will have one biomarker, and all of my bait station baits will have a separate biomarker. And this will come in handy for our pre and post bait trapping that occurs in July and October, where we collect blood and other biological data from these animals. And then the blood serum from all these captured animals will be analyzed for evidence of an rVNA response, as well as the biomarkers. Okay, so just a quick field update. I just finished up my bait station study, and by the conclusion of the study, 93% of the baits had been taken from the bait stations. And then we also just wrapped up our trapping session just 2 weeks ago, and we had a record number of captures in both July and October, which is pretty cool. So the data that's collected that's going to be used to develop a model of a raccoon being vaccinated as a function of one of two variables that fall within, or variables that fall within one of two categories, being those related to baits and those related to land cover. So each animal that's captured in the post-baiting period, so October, will have one of four outcomes listed there. And then I'm going to be developing a multinomial logistic regression model that predicts the probability of each of these outcomes. So this is going to allow me to identify the who, the what, and the why our serum conversion rates are lower in Burlington and hopefully other urban environments. This will also allow me to inform the NRMP on the best ORV strategies to maximize the probability of bait consumption by our target species. And I just want to take a moment to thank those listed on the slide and many more. It has really taken an army to get this project up and running, and couldn't have gotten this far without them. So thank you. Any questions? Yeah.
I don't know if you could tell individuals from the photos, but do you know of any raccoons that just like hit the same bait station over and over again, the same individuals?
So we actually don't have cameras on our baits in the bait station study. I would really love to, maybe next year, but I wouldn't be surprised if there would be, you know, like one or two raccoons that just sits there. But we'll see. That, I mean, my biomarkers will help tell us if that's the case or not. So, yeah.
Would that kill a raccoon over time? Like, can you over?
No.
Really dumb question.
No, it's not a dumb question. No, as far as we know, they are completely harmless. There have never been any adverse effects that I'm aware of using these on-rab vaccine baits. Yeah.
I had a similar kind of follow-up question about humans, actually. So a kid in particular.
Yeah.
Very dangerous.
Nope, there's using on-rab vaccines. Again, this is as far as I'm aware of, there have been no reports of this. Yeah, we have had people like call the Vermont Rabies hotline saying like their kid ate a bait or something. And, you know, usually they might have, we say they might have an upset stomach, but as far as we're aware, this is something that is going through the FDA. On-rab vaccine baits are still a trial, so they have not gone through the FDA quite yet, but the other vaccines have, so.
Will they not get rabies?
The kids, well, hopefully not. We try to tell people don't vaccinate your animals with these, you know, or your kids, but who knows if they listen. Yeah.
I guess do you have any like thoughts of why the uptake is lower, like personally, or like any inklings about why it might be? Yeah,
I think part of it is because they get thrown on a lot of people just try to get them out because it's such a huge operation and it takes so much time that in these urban areas where there's like so much sidewalk, they just like try to get them on any grass that they can. And a lot of the times it's like I've gotten pictures of like lawnmowers just chewing up these baits. So I think that's a possibility. And also, as I said, we can't really access all of this habitat, so nothing's confirmed yet, but I think that the bait stations will help because we can put those in targeted spots where we know that animals are going to be moving. Okay.
Any remaining questions from the audience?
One more question. So you showed a bunch of different versions or of the baits where they've been eaten partially.
Mhm.
Is there like if an animal eats like one bite of one and it's like slightly chewed, does that mean that it's vaccinated or does it eat the whole thing?
No, so honestly, even if they eat the whole thing, it doesn't necessarily mean that they're vaccinated. That's why we use we do the trapping efforts and collect blood, because it has to be, I don't know the exact number off the top of my head, but it does have to be over a certain amount in their blood for them to have this response. But the vaccines, you can like feel the liquid in them. So if they only chew the corner of it and there's no liquid exposed, then you can't, they definitely were not like vaccinated from that. But yeah, so I guess it depends really. Probably depends on the individual as well.
Thank you.
Yeah.
Wonderful.
Yeah.
Thank you, Maggie. Awesome. So our next presenter is going to be Lindsey Pekurny. Lindsey's interested in work that moves research forward into management action. Her research focuses on creating management tools that navigate the uncertainty surrounding management decisions in order to implement impactful management actions. And Lindsey's dream animal hybrid would be a red ect and a chickadee, poisonous and feisty. So Lindsey, you can take it away.
Hi, everybody. Okay, so before I get started, I'd like to just acknowledge that I am, my work is a collaboration between the U.S. Geological Survey and the University of Vermont. Within the USGS, I am working with NEARMI, Northeast Amphibian Research and Monitoring Initiative. So I just wanted to take a second and thank them. So my thesis focuses on managing for amphibians in complex ecosystems. I'm interested in creating management tools that help put management into action. Often we get stuck kind of just continually doing research and gathering more data and more data. However, species are declining and we really need to start taking action now. So I'm interested in making those tools that can get actions into place. So, as I said, the main objective for me is to create those management tools. My first chapter focuses on the occurrence of red-spotted newts throughout the Northeast. My second chapter looks at managing for multiple pathogens simultaneously within an ecosystem. So my first chapter focuses on, like I said, red-spotted newts. There's a lack of baseline data as to where these newts occur. So there we go. So this is the IUCN Red List range map for the newts. From this range map, we can't really tell like where do newts actually live in that giant orange blob. So my focus for this chapter is to figure out where the probability of occurrence of red-spotted newts throughout the Northeast section. So just up here. For anybody that doesn't know, red-spotted newts are those really cute orange guys you find out hiking. That's the juvenile stage. The adult stage is aquatic. My work is based off of data collected from adult aquatic newts. Oh, some threats. So amphibians are the most endangered group of vertebrates currently in the world. So there's a lot of threats affecting them: habitat degradation, invasive species, pet trade, disease. So the aim of my study is to figure out the covariates that are driving occupancy patterns of newts, as well as variables that are affecting our ability to detect newts. And using those covariates, I'm going to predict the occupancy patterns of red-spotted newts in the Northeast. So as I said, I've been collaborating with the USGS. The data that I'm using is from a study that was put on to survey for an emerging disease of newts. I use the R package RPresent to run my occupancy models. And for my covariates, we didn't specifically collect environmental data from the field, so I had to rely on expert opinion and literature. So my covariates that I did use, I have two types that have occupancy covariates, which are human interference, that is like roads, light pollution, habitat degradation, as well as, and then our detection covariates include method, type, and day of year. So my predicted results, we predicted that occupancy will decrease as human interference, that's the HII, increases. We also predict that newts will occur more so in deciduous forests versus coniferous forests. Salamanders in general need moist substrate to survive. So for detection, we predicted that detection would follow breeding activity, so highest detection during the months that the most breeding activity occurs. And we also predicted to find that detection would be higher when we were using traps versus dip net. So my results from this first chapter, human interference really influences patterns of newt occupancy. This map here is based on our human interference index. So occupancy, the brighter the color, the higher the probability of occupancy. For covariance, as the summer increased, we found that day of year negatively impacted the probability of detection. DipNet, we were surprised to find, had a higher probability of detecting newts than traps. We think that might be because dip netting, you're actually going to the pool, you're walking around with your dip net and you're actively searching for the newts, whereas traps, you just throw the trap in the pond and leave it there overnight. So the impact of this is a tool that can be used to understand where newts like to live within the Northeast. As I mentioned before, there's a lot of threats facing amphibians. Currently, there is a fungal disease in Europe that we expect will come here, and using this map, we'll be able to better prepare for where we might need to manage if it emerges here. So my second chapter is focused on managing for multiple pathogens simultaneously within an ecosystem, and in general, ecosystems are really complicated. There's a lot of interactions going on, whether between biotic or abiotic factors. And so the question I'm interested in is how can we efficiently make decisions in the face of uncertainty and with limited time and resources? So the objective is to create a management tool in the form of a Bayesian belief network to model interactions of pathogens within an ecosystem. So the two pathogens I'm interested in are BD and ranavirus. So I'll try to say it, Batrachotrytium dendrobatidis. They're both globally distributed and both responsible for mass amphibian die-offs. BD is a fungus, ranavirus is a virus. We're also interested in vernal pools in protected areas because that's where we have the jurisdiction to actually make management changes. So study system, we're focused on wood frogs and spotted salamanders. Their ranges overlap. Our study is in the southern portion of the wood frog range, the northern portion of the spotted salamanders. Both these guys breed in vernal pools. They're both susceptible to BD and ranavirus. However, they differ in their rates of decline and susceptibility to disease. Wood frogs are rather short-lived. They live for about 3 years. Spotted salamanders can live for 20 plus. So I think that might have some influence onto their decline. So the methods for this chapter, I'm still in the literature review portion of this. So the first step was to do a large lit review, kind of figure out all the different variables and interactions that might play a part within an ecosystem on these two pathogens. Then I refined my lit review to focus on 3 major variables. In this refinement stage, I was looking for environmental factors that might either enhance one pathogen and decrease the other, or vice versa, or something that causes an increase in both of them. And eventually I'll put this into a Bayesian belief network. They combine expert opinion with existing data. So this is where I'm at right now. I have temperature, salinity, and vegetation. These are 3 environmental factors that I believe we could actually manage for. The other guys down here are also important, but they'll be a little bit more difficult to manage for. So all 3 of these affect the pathogens in opposing ways. The next step will be to assign probabilities of, well, first to figure out some management actions. So this is an example of a BBN. What we have here is management actions, how those actions affect the specific variable you're looking at, and how doing that action affects the pathogen that you're looking for. So taking the temperature, salinity, and vegetation, figuring out probabilities and different management actions for them, and how that would influence the ecosystem and therefore the pathogen is my next step. So the impact of this is going to simplify decision-making for complex systems. Like I said, it'll take some of the uncertainty out of decision-making and hopefully help get management into action instead of getting bogged down and continually doing more and more research. So overall, managing ecosystems is hard, probably for anything you're studying, but we really need to be able to make efficient management decisions, especially as we're running out of time to save a lot of species. So combining current knowledge with new modeling tools can give us a way to kind of get to the action bit a little bit quicker than if we didn't have such tools. Okay, with that, I'll take any questions.
For your first chapter, how many sites did you have?
264.
You collected all of the data?
Oh, so it was collected over 2016 to 2017, so it was a previous data set. I've been working with the USGS, so I was part of that team, but it scaled from like Virginia to Vermont, so we had multiple teams collecting data. So it was a pretty big effort.
Did you find that the pathogens were dispersed evenly throughout the different regions that you were studying, or was one favored in the Northeast versus like the South?
So this specific study was actually surveying for an emerging bee sow. It was a sister pathogen to BD. We didn't find any bee sow. Bee sow is currently in Europe, but our newts have shown to be 100% mortality rates in labs. So the study was designed specifically to look for bee sow, but other studies that the USGS has done, sorry, my mouth is getting dry, have found that ranavirus and BD are endemic throughout the Northeast. So they're here. It's just figuring out how to manage for them and how to, now that they're here, like what are we gonna do about it? That's why my first chapter I'm hoping can be used to prevent bee sow from establishing itself in the Northeast. So I'm hoping that these tools can be used for a proactive instead of reactive approach.
I just asked, did your like HII interactions index include trails, or was that just roads and stuff?
It had like 14 different layers in it, but I don't believe it included recreation, which is something that I would also be interested in. There is a paper that was done in the Adirondacks that looked at the of like do hikers spread BD, and I think their results were inconclusive, or they didn't find any correlation there.
On your new distribution map, I may have missed this, but you had the human appearance index. It was kind of that was the only factor you showed on that.
That was the only factor in that map.
Is it because it's the most important that you found, or are there other factors that you think are important?
Forest type, I think it also ranks highly in our model selection, but not human interference seems to be the highest ranked. Our second model was forest type and human interference. When we mapped them together, the map didn't look very different from the original HII. Like I said, the original study, the intent wasn't to determine these covariates. So if I were to redesign the study, I would certainly go back and I would wonder about factors such as salinity, definitely forest type, wetland type, water temperature, but we just didn't have that data. So I was relying on GIS data to.
Great, thanks.
Just going back to the 3 factors you were talking about that we could potentially manage with, what, I guess, what specifically would that entail? Like, how exactly would you go about managing for like salinity or temperature?
So I haven't thought too much about it, but I can. So salinity, I'm assuming that we could probably just go to a site and dump some salt in there. And then temperature, we can remove canopy. So BD does better in cooler temperatures. So one management practice that's been suggested is removing canopy above pools to increase sun exposure. However, if we did that, ranavirus does better with those warmer temperatures. So if we clear out the canopy, we just might be making the environment better for ranavirus. With salinity tends to kill BD. It's a fungus, so it just doesn't thrive in high salinity environments. Whereas ranavirus doesn't seem like the effect with ranavirus is almost synergistic. So tadpoles and frogs can survive in pools with high salinity, but if you add ranavirus to that, the effect is like higher ranavirus infection rates than if we had just an average pool with no salinity in a salinity pool.
So it occurs to me to ask, as we start trying to control runoff of road salt from roadways, is that going to increase the risk of the spread of these diseases?
So that's something I'm very interested in. After like kind of going through this literature, I'm wondering, it seems that urban areas might provide refuge for amphibians from BD. So you see like the little pool on the side of the road covered in trash. That could potentially be a refuge from disease for amphibians. However, that pool is probably more hospitable to ranavirus. So what I would be interested in studying further is how often does ranavirus occur in urban environments, and can we use urban environments as like areas of refuge for amphibians? I don't know the answers, but that's something that gets me excited to think about.
Very cool.
One more. Yeah. So I guess and I don't know anything about amphibians really, but my conception of amphibians is that they're an indicator species and that they're really sensitive to pollution. So when you're talking about using urban centers, that's kind of contrary to what I understand, because I would imagine that there's a lot more pollution, so it would be harder to make those places safer.
That's what's so fascinating to me. I think it's going to come down to the species as well as, and this is something that we could put into a BBN because it's like an interactive tool, and you can change the probabilities of what if I tweak this and how will it affect this. So there's going to be a trade-off between how much pollution can an amphibian withstand versus how much BD can it live with. And I think figuring out that like fine area is where Bayesian belief network might be most helpful. Yeah.
Well, thank you, Lindsey. All right, and last but not least, we have Matt Lacey, who will be speaking to us remote via Teams. So I think Caitlin's working on getting him up. But Matt is from Columbus, Ohio, received his Bachelor's of Science in Wildlife Conservation from Virginia Tech in 2018. He is now pursuing the UVM VLS dual degree program, so with the Vermont Law School, and has met all of his course requirements and is now just working on finishing his graduate research remotely. So he hopes to finish early next year, and it looks like we have him up and ready to go. And I guess, last but not least, if Matt could be the hybrid of any two animals, he would be a hybrid of a harpy eagle and a western lowland gorilla, because he loves to be able to fly, but also have the smarts and strengths of a gorilla. So, Matt, take it away.
And I'm going to be clicking through Matt's slides for him. You won't see him while he's talking, but after the presentation's done, I can bring him up in our little producer view and you'll be able to see him. So Matt, can you hear me okay?
Yeah, can you hear me?
Yes, we can. I'm just going to turn our speakers around. That might help a little.
Lindsey's presentation was a little jumpy on my end, so please let me know if I'm like cutting out. I closed some background applications. Hopefully that'll help.
Can you all hear him okay? All right, you're good to go. So you just say next slide when you want me to change slides.
Cool. Yeah, that was my issue yesterday. I kept forgetting. Great. So as Abby said, my name is Matt Lacey. I'm in the Vermont Law School, University of Vermont dual degree program. Here at the University of Vermont, I'm being advised by Dr. Jed Murdoch. Thanks for bearing with me to the end of the presentations, and thanks for the opportunity to present virtually. My graduate research is conducting a policy gap analysis of wildlife conservation laws and regulatory structures in Vermont. Next slide. So why analyze wildlife policy in Vermont? Vermont's currently undergoing a dramatic period of land use change. From 2012 to 2017, the state lost over 100,000 acres of forest land. The Vermont Fish and Wildlife Department estimates that the state loses almost 5,000 acres every year to regulated development alone. Further complicating matters, 80% of the forest land in the state is privately owned, making large landscape coordinated conservation efforts quite difficult. During 2020, we saw a spike in out-of-state property purchases, probably due in part to the pandemic. You can see that in the graph at the left. Next slide. Vermont was successful in restoring several emblematic species, including the moose, the common loon, the bald eagle, the wild turkey, and white-tailed deer. However, many are now declining as a result of new threats. Habitat loss threatens many grass and bird species such as the grasshopper sparrow and the golden-winged warbler. Utility-scale renewable energy development threatens black bears, the Bicknell's thrush, and several species of bat. Land use change contributes to habitat loss, increased pollution, and the proliferation of invasive species. Additionally, here in Vermont, we can expect to see a rise in temperature of 5 to 9 degrees by the end of this century. The climate crisis creates new risks for moose, Canada lynx, brook trout, and many others. Further, a changing climate will dramatically rearrange the distribution of natural communities in the state. Next slide. In addition to current trends in land use change and the threat of climate change, Vermont's wildlife policies are ripe for review due to the significance of the state within the greater context of New England. Vermont contains 5 high-priority wildlife linkages as identified by the Staying Connected initiative. We're just beginning to understand the regional importance of Vermont, and particularly in the face of a changing climate, this heightens the need for more integrated wildlife conservation policies. Next slide. So for this research, I reviewed 6 state laws that have significant implications for the conservation of terrestrial wildlife in the state. The 4 regulatory policies I reviewed include Act 250, Section 248, the Vermont Endangered Species Law, and the Vermont Wetlands Rules. In Act 250, I focused primarily on Criterion 8A, which states that development or subdivision can't be granted if it will destroy or significantly imperil necessary wildlife habitat or any endangered species. Under Section 248, the Vermont Public Utilities Council issues certificates for public good for certain energy and communication projects. During review, due consideration is given to Act 250 criteria, including 8A. And additionally, through this program, the Vermont Fish and Wildlife Department reviews timber harvest notifications for several biomass plants. Under the Vermont Endangered Species Law, the commissioner, based on recommendations from the Endangered Species Committee, can choose to list species as endangered or threatened and can now designate critical habitat. Due to the Vermont wetlands rules, all wetlands in the state are classified as Class 1, 2, or 3, and different levels of classification have greater protections or buffer sizes, and permits are required for certain uses. Next slide. The non-regulatory policies I reviewed include Act 171 and Vermont's current use program. Act 171 amended the state planning statutes to require planning for forest integrity at municipal and regional levels. Current use or use value appraisal is a tax incentive program for forest landowners to keep and manage private forests. 2 of the forest land categories that may be enrolled in this program are significant wildlife habitat and the ecologically significant treatment areas, which include rare, threatened, and endangered species habitat and vernal pools. Next slide. Many of Vermont's wildlife conservation policies were signed into law 30 to 50 years ago, and since their enactment, we have witnessed significant scientific advancements. The current suite of wildlife protections in the state was born out of a piecemeal approach, and while effective at several levels, is less integrated, which has led to management and conservation gaps. There's not been a comprehensive study of Vermont's wildlife laws across levels, which could inform current policy directions. Recent legislative initiatives, including the 2018 Commission on Act 250 and the enactment of the Global Warming Solutions Act in 2020, show a statewide desire for updating environmental laws. Next slide. Some gaps I predict may surface through my review include potential game species bias. Since 2009, approximately half of the habitat protected by Vermont Fish and Wildlife through Act 250 and Section 248 regulatory review have been deer wintering habitat. Additionally, staying on Act 250, jurisdictional triggers may provide another loophole because Act 250 jurisdiction only applies to subdivision developments above a 10-acre or 10-unit threshold. Looking at the Vermont wetlands rules, there may be gaps related to the allowed uses which could have detrimental effects to wildlife. Next slide. My hypothesis is that current environmental laws in Vermont only benefit a fraction of wildlife species in need of conservation, and many of the current threats to wildlife and wildlife habitat are unaddressed in the current legal system. My goal is to identify the gaps in the current regulatory structure and assess the impact of those gaps on the management and conservation needs of wildlife in Vermont. Next slide. As a first step in my research process, I reviewed each law. To standardize this review, I utilized NAJM's 5C of policies protocol described in their 1995 paper. Next slide. So the 5 C's of policy are content, context, commitment, capacity, and coalitions, where content is the goals and methods of the policy, context is the operating procedures of the implementation of the policy and any boundaries to implementation, commitment is the implementing party of the policy, capacity is the administrative capacity of the implementing party, and coalitions refers to those whose interests are enhanced or threatened by the policy. Next slide. To identify the policy gaps in Vermont's suite of wildlife policies, I sought to create a novel methodology that was both objective and replicable. I utilized and modified the methodology from the paper described on the next slide. So next slide. In their 2012 paper, Understanding Policy Coherence, Nilsson et al. Designed a policy coherence study that utilized an illustrative screening matrix, where screening was conducted as a rapid assessment in an in-person workshop of environmental experts, where experts scored interactions between policies as strong, weak, or neutral. This study looked at both vertical and horizontal policy coherence, where vertical coherence or vertical policy refers to levels of government such as local, state, federal, and international in this case, and horizontal would be across sectors, so say environmental, energy, agricultural. Next slide. Working off this screening matrix, I designed a survey using Qualtrics focused primarily on intersectoral policy coherence with some horizontal applications such as forestry and agriculture. I drafted 23 conservation objectives that could be broadly grouped into the following categories of wildlife, wetland habitat, forest habitat, rare, threatened, and endangered species, and general. These objectives were drawn from the statutes, rules, and guidance from each of the 6 policies. In the survey, respondents will be asked to describe the relationship between the implementation of the policy and each conservation objective. Following the matrix, they'll have the opportunity to elaborate in a short answer section and describe any gaps related to capacity, any other policy gaps, trade-offs between policies, and proposed policy solutions. About a week and a half ago, I sent this survey out to 63 wildlife conservation policy professionals in Vermont. Next slide. Here are a few screen captures from the survey. On the left, you can see the policy coherence matrix. Bottom center, you can see at the beginning of each matrix, I asked respondents to describe their familiarity with each law. We don't expect everybody to be a subject matter expert on each of the 6 laws. And within the matrix, they were able to select unknown as an answer as well. Next slide. Following my gap analysis, I plan to review other states and other federal wildlife policies different from those here in Vermont. I will partially utilize NAJM's framework, focusing primarily on content, context, and coalitions. Next slide. Bringing everything together using my preliminary review of each existing law in Vermont, the policy gap analysis, and my review of wildlife policies outside of Vermont, I hope to propose 2 to 3 new policies to address the identified gaps. I would like these solutions to be relevant to policymakers and will likely steer away from climate-related policies because of the ongoing work of the Vermont Climate Council and my own personal expertise in land use policy, wildlife policy, and forest-related policy. Next slide. I'd like to acknowledge my advisor, Dr. Jed Murdoch, my committee members, Dr. Alan Strong, Dr. Walter Pullman, the Rubenstein School of Environment and Natural Resources, the Vermont Law School, the Heiser Graduate Award for supporting this research, all those who have taken the time to respond to my survey, and those who have yet to do so, but hopefully will, and all the others listed who contributed to the development of my project. Next slide. Here are my references. Next slide. And with that, I'd like to open it up for questions. Thanks for listening.
All right, so this is going to be a little funky for those watching online, but we're going to stop sharing. And then let's see. And that should pop up. Oh, I got to pull them up over here though. Let's see how that goes.
We don't have to show my face.
We want to see you. You're dressed so nice today. Let's see. Oh, there he is. All right, you're on a projector now, so we can all see you.
So any questions for Matt, anyone?
How can we fill out your survey if we didn't get it somehow?
You could send me an email and I'd be happy to send you a personalized link. We're sending out personalized links so that people aren't sharing it. We want to have some control over the sample, but if you think that you could contribute something valuable, which I'm sure that you could, please shoot me an email. I can send Caitlin, or Caitlin knows my email. Oh.
Yeah, that was your co-presenter, Stever, so I can send you emails.
Cool.
So you talked about there potentially being biases in what's being managed right now, like for game species, but then you said you want to identify gaps in the policies and assess for wildlife. I'm wondering if you have certain wildlife species in mind, assuming they're non-game species, and because it seems like that would be a difficult thing to do for all other species that aren't the ones that are already biased for. Does that make sense?
So you're asking what species I expect to see a bias of management efforts towards?
No, the other way around. Like, you're trying to identify gaps in what is being focused on. So I'm just wondering if you have in mind certain species of interest that you are going to try to think about when you're identifying the gaps, because you wanted to have assess it for wildlife management reasons, I guess.
Yeah, that's a great question. So I think I quickly slid this in, but I am primarily focusing on terrestrial wildlife just because of my professional background and scientific understanding. At the beginning of the presentation, I mentioned a couple of grassland bird species that are in decline. I listened in on another presentation that was talking about renewable energy development and solar development. I think could be one area where there might be conflicting objectives and goals, where there could be a gap. Unfortunately, to keep my survey standardized and kind of broad, higher level, I didn't ask too many species specific questions. So I'm not sure that my results will show or point directly towards any particular species. Does that answer your question?
Yeah, thank you.
Any remaining questions from the audience?
Matt, you indicated that your intention was to develop several policies, and I forget the other category, but so if you develop those, how would you expect to be able to introduce them? I mean, would you advocate to introduce them or what?
I mean, I hope to publish my research. The central theme in the publication would probably be on this novel policy gap analysis and less focus on the solutions. But I will be writing up the solutions as part of my project. I would love the opportunity to speak in front of the Vermont legislature, or share my research findings and any papers or written components with legislators.
Thank you.
Wonderful. So it's just past 7 o'clock. Any remaining questions that you're itching to ask, Matt? Okay, well, thank you, Matt. And congratulations to all the presenters. You guys were awesome. Thanks for such a great graduate research symposium. Please join us in the solarium for snacks and cash bar. And we hope we can all see you there. Unfortunately, Matt will not be there, but yeah, so we can all head out there. And thank you for coming. And thank you to Caitlin for being our techie.
Thanks to everybody online.
I ordered a lot of free food for you all. Please eat it. The alcohol unfortunately is not free, but if you have cash, that's okay. There's that.
Yes.